So cell cycle deals with the total changes between one cell division to another cell division. And the details of the cell cycle was given by the scientists named Howard and Bailey in 1953. And let's start with the interphase. And the interphase is the first phase or first stage of karyokinesis. That's the division of nucleus. It is a stage of the gap between the end of one mitosis division to the starting of next mitosis cell division. This is the metabolically active phase, which is also known as the preparation phase. So one of the objective questions you may be asked is, which of the following is called as the preparation phase? Then your answer will be interphase. Likewise, it is also the metabolically active phase. And it has been divided into subphases. So the first phase here is the G1 or the gap 1 phase, which is also called as the post-mitosis phase. So let's see what happens in this phase. Here the cell grows in size. There is cell growth, where there is the synthesis of protein, carbohydrate and lipid. Chromosome becomes long and thread-like. That is, the chromatin fibers in this phase are less square and slender, extended fully and ready for transcription. And the nucleus and the cell become enlarged here. Likewise, if it has to check for any damage or errors in its DNA, and repairs them if possible, and if the cell is not ready to enter in the next phase, then it can enter a resting state that is called as the G0 phase. And the chromatin, as it is ready for the transcription, the process of transcription results in the production of 3 types of RNA, and the G1 phase is the linear phase than the other phases. So, and the second one is the S phase, which is also known as the synthesis phase. Synthetic phase, where their synthesis of histone protein takes place. Histone protein synthesis takes place. Likewise, there is a duplication of chromosome number takes place. Likewise, it is the place where there is active DNA synthesis, DNA replication takes place, and it occupies 35 to 45% of the cell cycle. Now let's move towards the another phase, that is the G2 phase or the gap 2 phase. Let's see here, the gap 2 phase is also called as the pre-mitosis phase. In this phase, the changes that we see is here they place the synthesis of 3 types of RNA takes place, and the and protein also. There is synthesis of protein, which is for the cell growth and for the duplication of the cell organelles. It occupies 10 to 20% of the cell cycle. It occupies 10 to 20% of the cell cycle here, and at the G2 phase draws to a close, the cell enters the M phase. The M phase is known as the mitotic phase, and cell divides into 2 daughter cells. The M phase consists of the 2 subphases, that is the mitosis and the cytokinesis. Is the cell division second type after the mitosis cell division, which is also known as the indirect cell division. Indirect cell division. The name mitosis was first given by Fleming. The name was first given by the Fleming, and it is also known as the equational cell division. So this may be your objective question, which of the following is also known as the equational cell division? Then your answer will be mitosis cell division. It is also known as the somatic cell division. It is also known as the somatic cell division, as it occurs in somatic cell, and it is also known as the also called as vegetative cell, which has called a somatic cell division, and it is present in the vegetative cell, which helps in the growth and development of cell, as it is present in the vegetative cell. Also called as the somatic cell division, occurs in the somatic cell as present in the vegetative cells. It occurs in the leaf tips, shoot tips. So this is your next important objective question. Where does the mitosis occur? It occurs in the leaf tips and shoot tips. Got it? Important for the wound healing and generation of new cells. It is not involved in sexual reproduction. There is the formation of diploid cells. So first of all, it has been divided into three major phases. And the first phase is obviously the interphase. It is the stage between the end of one cell division to the beginning of another cell division, and during this phase, nucleus and the cell become enlarged, replication of DNA takes place, duplication of cell organelles, nuclear membrane and nucleolus are well developed, and so on. And the second phase is the karyokinesis, that is the process of division of nucleus, and it completes in the 4 phases. Okay, let's continue here. And the first phase is the prophase, which is obviously the longest phase. And this is an important question, that which of the following phase is the longest phase in the mitosis? Then your answer will be prophase. In this phase, in case of, let's talk about first of all, in case of the early prophase, there is the nuclear membrane and the nucleus are well developed, and obviously the chromatin fiber is also quite visible in case of early prophase. And in case of the late prophase, the nuclear membrane and the nucleolus start to disappear. In late prophase, it starts to disappear, and the formation of the spindle fiber takes place. And the formation of the spindle fiber takes place. This is the spindle fiber. So this is the case of early prophase and the late prophase. It is a state where the nucleus starts to disappear and the spindle fibers are formed, and DNA condensed into the sister chromatids and held together by a special DNA containing region, that is the centromere, which is also known as the primary constriction. So here is another important thing that you have to know, guys, that is the centromere is also known as the primary constriction. Centromere is also known as the primary constriction. Now let's move to our next phase, that is metaphase. And the metaphase, in this there is the disappearance of the nuclear membrane and nucleolus completely, and there is the formation of spindle fibers. You can see the spindle fibers here. Its formations is completed, and the chromosomes are longitudinally divided but are attached together by the centromere, single centromere. There is single centromere which is attaching the sister longitudinal divide chromosomes, and all the chromosomes are arranged at the equator of the cell to form the metaphasic plate. Here everything is just arranged at the equator. So in which of the following phase the metaphasic plate is formed? Then your phase answer will be metaphase. And the process of the chromosome lying in the equator is known as congregation. Another important thing is that the chromosome lying in the equator is known as the congregation. So this is also an objective question. And another thing is that it is the base stage for counting the number, shape and size of the chromosome. And you can see in this picture that the arms are, in case of metaphase, are towards the pole and the centromere is at the center. And equatorial metaphasic plate is formed. Metaphasic or equatorial plate is formed and the sister chromatids orient alongside the cell equator by linking their centromere to the spindle fiber. So this is the case of metaphase. Now let's move to the another phase, that is anaphase. It is the phase which is the shortest phase. In case of longest phase it was prophase, and in case of shortest phase it is the anaphase. Here this is also a part where the also known as the shortest phase, also known as the migration phase, and the spindle fiber here contracts and divides the centromere and pulls the chromosome towards the respective poles, and the centromere of each chromosome divides longitudinally. As a result, the two sister chromatids have their own centromere after the division, and in this way half of the chromatids move to one pole and half of the chromatids move to another pole. And the chromatids here during this pole wall migration of daughter chromosome, they appear in different shapes like V, U, L and J shaped, and the arms here are projected towards the center and the centromere is projected as towards the pole, just opposite to the metaphase I have already shown you. So this is the case of anaphase. And let's move to another phase, that is the last phase, that is telophase. It is also the visible phase and also called as the reorganization phase. It is called as the reorganization phase because everything it becomes at the start. It reverses itself, where the daughter chromosomes reach to their respective pole and the chromosome absorb the water and it begins to uncoil and forms its previous thread-like structure, that is the chromatin fiber, and the spindle fibers disappears and the nuclear membrane and the nucleolus starts to appear, and the two opposite chromosomal sets are formed, and at the end the two daughter chromosomes are formed, and each nucleus contains the same number of chromosomes as the mother cell. So it is also known as the equational cell division, as mentioned at the first, that the mitosis is also known as the equational cell division because the two daughter chromosomes or the each nucleus contains the same number of chromosomes as in the mother cell. It is also known as the reorganization phase. Here the spindle fiber disappears, nucleolus and the nuclear membrane reappears, resurfaces and resumes their long, extended, slender chromatin fiber appearance. And this is the image of the telophase. And now let's move towards the last stage, that is the cytokinesis. After the completion of division of the nucleus with the four phases inside the karyokinesis, now move towards the cytokinesis, which is called as the division of cytoplasm. That is after karyokinesis, and it takes place through two ways. The first one is obviously the cell plate formation in case of the plant cell, and the another is by furrow formation or by the constriction method, also known as furrow formation. And this is the cell plate formation, where in case of this the cell membrane splits, animal cells drift away and the plant cells form a cell plate which turns into a new cell wall. Got it? It turns into new cell wall. The plate, in case of the cell plate formation, this type of cell division is found in the higher plants, where the small droplets of the granules, which is known as the phragmoplast, which are produced by the Golgi bodies or endoplasmic reticulum, and they later become united to form a plate-like structure which is called as the cell plate. So these are all the phragmoplasts which have been released by the Golgi bodies or the endoplasmic reticulum, and later it forms the cell plate, or it's also called as the medial lamella. And next one is by the furrow formation or the constriction method, and this type of cytokinesis takes place in the animal cell, that is the lower plant cell and the bacteria, and in this process the cytoconstriction appears at the middle of the cell. There appears the constriction at the middle of the cell and divide to form the mother cell into the two daughter cells by the furrow or the constriction. It is essential for the growth, development and maintenance of the living organism. It plays a role in the tissue repair, wound healing, and immune response. Gonads at the sections depend on the mitosis for increasing in the number and maintenance of equilibrium in the amount of DNA and RNA in the cell. Now let's see shortly what happens in mitosis. In case of the early prophase, the nucleolus and the nuclear membrane start to disappear, and the chromatin fiber is visible in case of early prophase, and in this phase there is the disappearance of the nuclear membrane and nucleolus, and the formation of spindle fiber starts. And in case of the metaphase, the disappearance of the nuclear membrane and nucleolus and the formation of the spindle fiber gets completed, and the chromosomes appears lying in the equator, and this is called as the congregation, and there is the formation of metaphasic plate or the equational metaphasic plate in the metaphase. And in case of the anaphase, first the arms are towards the pole and the centromere is at the center in case of metaphase, and where in case of the anaphase is just opposite, that the centromere is towards the pole and the arms towards the center. And in this phase, this is the most shortest phase, also called as the migration phase, and prophase is the longest phase obviously, and this is the shortest phase, also known as the migration phase in case of anaphase. And in case of telophase, it is the reorganization phase, also called as the reorganization phase, where the chromosomes absorb water and begin to uncoil and form the chromatin fiber, and the spindle fiber disappears. The nuclear membrane and the nucleolus again appears, and at the end the two daughter cell chromosomes are formed. So this was for today. Thanks for watching. If there is any query, then please comment down below.